Good morning. I'm Dr. Gemma Ma. I'm a veterinarian from RSPCA New South Wales, and I'm one of the animal welfare representatives on the Animal Research Review panel. I'll be your host for the webinar today. I'd like to begin by acknowledging the traditional custodians of the various lands on which we meet today. I pay my respects to elders, past and present, and recognise and celebrate the diversity of Aboriginal peoples and their ongoing culture and connections to land, sea, and community. I also extend my respect to any Aboriginal or Torres Strait Islander people joining us today. Thank you everyone for joining us this morning. I'd like to welcome you all to our second Animal Research Review panel webinar for the year. Today's theme is housing and enrichment for animals in research settings, and we've got three wonderful speakers for you. Before we get started with our first speaker, let me run through a couple of housekeeping points. Firstly, the webinar is being recorded. If you have any questions for any of our speakers, please submit these as we go. You can do that through the Q&A tab. We'll get to questions at the end of the session after we hear from our third speaker. We'll answer as many questions as we have time for. Any questions that we don't get to today, we'll circulate a Q&A document following the webinar, so you won't miss out. All right, well, let's get started. First up, I'd like to introduce Dr. Findy Wills. Findy is a veterinarian with a master's degree in clinical animal behaviour from the University of Edinburgh. Findy started her veterinary career as a small animal vet in private practice in Newcastle, New South Wales, before starting work with shelter animals at RSPCA Sydney in 2015. In 2020, she started her private practice, Let's Talk Pause Veterinary Behaviour Services, to provide behaviour consultancy services to clients and animals between the Central Coast and Newcastle. In the same year, she started offering her consultancy services for RSPCA New South Wales, with her main focus being on providing post-adoption support for shelter animals. Findy is passionate about advocating for human-animal relationships and is looking forward to conducting further research in emotional regulation in animals. Over to you, Findy.
Hello. Can everyone see me? Hear me? I hope so. I'll just share my screen.
Yes, you're good to go.
Excellent. All right. So I'm Dr. Findy, and today I will be talking about housing and enrichment as tools for emotional management of dogs housed in research facilities. So just in case, I'll just go through this quite quickly. I assume most of you will have some knowledge in this. So just a brief introduction. So the use of dogs in research, there's 5 main areas that we use dogs in research. So there's biomedical research, where we use dogs as models for human diseases. There's product development, product safety testing, pet food testing, and diagnostic studies. And according to the New South Wales 2021 Animal Use in Research Statistics Report, there's about 886 dogs currently used in research but are not privately owned. So these dogs won't be staying in a home environment. There's not a lot of public information about the housing environment of these dogs, unfortunately, but it would be assumed that they would be housed in kennels with access to runs and yards and, in line with guidelines. Now, how long are these dogs kept for? So according to a recent amendment to the Animal Research Act 1985 and the Animal Research Regulation 2021, 3 years is usually when they come into a conclusion for their use in research, and these dogs or cats will be looking at taking reasonable steps to be rehomed. So because there are so many animals, about 800, kept for 3 years within a research environment, it is quite important, and it's an obligation for us to maintain good welfare when they're under your care or our care, mainly to make sure they're of good health, that they're physically and emotionally free of pain and disease. The other quite important thing is, because we are using them for research, by protecting their welfare and making sure that they're not mentally or physically stressed, we're actually ensuring the validity and accuracy of these research results. Which brings us to the next point, where because the primary goal is really just trying to protect their welfare for the duration of their stay and the purpose of being in research, it's quite important for us to protect their welfare by providing physical and emotional needs, meeting them quite well. My particular area of interest is in animals' emotional health. I think most of us are quite well rehearsed, especially people who work with animals. Most of them would know how to monitor, flag for physical disease and illness, whereas emotional health is a little bit newer in this kind of health arena. So it is one of my areas of interest. So I will be predominantly talking about this component for this talk. So the two main components that support emotional health, what we need to understand, the two main things would be how to meet an animal's emotional needs and help them regulate these emotions. And by understanding emotional regulation, we'll be able to learn what we can help them, how we can manage and help them regulate their emotions with resources that we have. So these are the two key points we'll be talking about. So I'll be going through emotional needs and regulation of a dog first, so everyone understands what's required for them, and then how we can apply housing and enrichment, particularly for this purpose, for emotional management for these dogs to maintain their emotional health. So first off is, before we understand fully emotional regulation, we have to know what emotions they will encounter. In dogs, we use Panksepp's emotional brain models. He's a neuroscientist that mapped out circuits in the brain that correlate to emotions that dogs will feel or animals will feel. So we'll just apply it to dogs, which is pretty handy. So there's 8 that we talk about. We've added, so Panksepp's model is generally just 7, but in veterinary behaviour, we've added an extra one, or we separated one of them to make it 8, so we can focus on 8 instead of just 7. So we'll go through each one of them. Fear and anxiety is the first one, and everyone's quite familiar with. So fear is what you're scared of now. Anxiety is what you're worried about, so the what if questions. When the brain goes, oh, I feel there's a bit of fear and anxiety going on, what it's trying to tell the creature or us to do is to increase space. So we have more space to kind of observe the situation, feel safe, etc. And also to reduce interactions with what we perceive as a threat. So that's actually quite important to remember as a motivation when they feel this emotion. The second emotion we talk about is frustration and rage. So frustration is an emotion that fuels motivation. So if you want to reach a goal and you can't get there, something's stopping you, this is where this emotion comes to play. You go, you can do it. Let's make it happen. And what happens is when you feel frustrated, it's actually, again, it's a fuel. So it increases the intensity of your behavior response, and it increases the speed of that behavior. So it's often, sometimes you can get general primary frustration depending on the life stage that you're going through. Teenagers, generally, if you remember teenager age, that's the primary emotion that comes with it. Quite often it's also a secondary emotional need that comes in when any other emotional needs are unmet. So for example, if you have an anxious dog towards, say, humans, so the motivation for this dog would be making space around it between the human and itself, and reducing the interactions. If over time it can't accomplish this goal with whatever strategies it's trying to use, then frustration will come into play. So then you will see more intense behavior. So you can get more, say, barking or behaviors that are associated with what we associate with aggression, where they repulse the threat. So they bark at the human, or they try and growl at the human just to make space. That's when usually the frustration component has come into play to try and achieve the goal of making space. So that's an example of frustration. Then we have panic and grief, which is to do with attachment with caregivers. Pain is the one that we've added on as veterinarians because we would like to focus on this area primarily because dogs and cats don't say ow when they first feel pain. They can definitely vocalize, but usually the first thing they show us is by behavior. That's why we listed it there, just so everyone's aware that sometimes when behavior doesn't particularly make sense or there's conflict, this is an emotional system we should be thinking about. Then we have seeking. So seeking is an emotion where you're looking for information. For dogs particularly, it would be sniffing or coming over and looking for social interaction with you or with other dogs or animals, etc. There's quite a lovely, engaging emotion towards the environment. You've got lust and care. That's for your entire animals when it's to do with reproduction. Then you've got play, which is social play, interacting with each other. So they're quite often in any context when we talk about emotion and emotional needs in dogs or non-human animals. They're the 8 systems we generally always think about. So why they're in different colours is because they have different purposes, per se. Your red colours, they're what we group them as protective bias emotions. So when they come in, it's because the animal is perceiving a threat. So it's based on survival. So these emotions come in quite large volume and quite fast because the brain wants you to act quickly to get things sorted so you will not die. So they're quite high arousal is what we'll call. I'll touch on that a little bit later as well. Then the green ones are what we classify as engaging bias emotions. They're the ones where the brain goes, oh, these are great things. You do more of that when you feel this because we want you to thrive and survive. So they're a little bit more lower arousal emotions compared to your protective bias emotions. Now we understand or have a little bit of an idea what emotions that we can encounter. Then we'll talk about how they are regulated in a non-human animal. And this concept is particularly important for emotional health. When an animal cannot regulate its emotions, it's emotional dysregulation, and that's when you usually get quite intense and inappropriate behaviours that are quite hard to handle as a caregiver. So understanding how to help them regulate and provide effective management would be one of the most important things as caregivers to maintaining a creature's emotional health. So this is the next bit we'll dive on into is emotional regulation. So with emotional regulation, we use Sarah Heath's SYNC model to demonstrate this concept. The SYNC is an illustration of how much emotions you can hold, your emotional capacity, essentially. And the whole goal of emotional regulation is to regulate this SYNC capacity. So we don't ever want the SYNC to flood. So when the SYNC's flooding is what we usually associate with, oh, this dog is quite overwhelmed or it's quite stressed. Or any behavior that you go, oh, that's a bit extreme and out of context. It's usually to do with dysregulation. So there's 2 components that would control this SYNC. And if we understand what influences the SYNC, then when we apply strategies, particularly with housing and enrichment, it's just easier to understand the purpose of each one and how we use it. Then your outcome would be a little bit more purposeful and more effective. So first thing would be the taps, and this is where our emotions come in. So with taps, there's 2 things. One, it's like a hot and cold tap. So this will be your the red one. So the protective bias emotions, so your fear and anxiety, frustration, panic and grief, pain, they would be what we classify as the hot tap. So when that comes running, everything's quite hot and fast. So you can get dysregulation a lot quicker compared to your cold tap, which is your engaging bias emotions, seeking and play and all that. So they're your 2 taps. When there is another thing that happens is when you have both taps running at the same time. So that's when you get mixed emotions. That's mixed conflict is what we classify that as. And usually when you have a conflict of emotions, the volume of emotions that come down is also higher. And I'll touch on that a little bit later as well. So the second thing we talk about in terms of TAPS is arousal. So arousal is the volume and how fast things come down. So they are related, but they're a little bit different. So always think about, oh, in this context, if the dog is doing this thing, we go, oh, what emotions is it encountering? And what's the arousal of these emotions?
Have you played another game?
Then drainage is the other thing that can influence the sink capacity. So drainage is, we'll talk about a few things in drainage. The components would be decompression and recovery. Sleep is very important. It's one of the most important things that help with the drainage of the sink. In dogs, we would really like them to sleep for about 16 to 18 hours. For an adult dog, for puppies, I don't know if they have a lot of puppies in facilities for research, but you really want about 20 hours for puppies. And then for more senior dogs, it goes up as well to more than 18, depending to their needs. So apart from sleep, we usually say decompression activities are quite important as well that help the emotions drain. So it's equivalent of us, say, doing mindful activity, reading, colouring, going for a walk, etc. So for dogs, because they're scavengers, sniffing, chewing and licking are quite decompressive exercises for them, and we generally like to make sure that they have opportunities to do so in the day. The third thing is resilience. So relationships and social support is also quite important for an animal's recovery, and it's one of those things that can really help with emotional regulation if you have that support system with you. So in relevance to dogs that are kept in confinement and in kennels, these are the challenges they go through. So I'll go through these ones and then I'll try and apply this sync model so you can understand in terms of regulation, what are the challenges for these dogs? And then the next component with how housing and enrichment can help with that. So in kenneled dogs, there's four main things that we found that they are really usually struggling with. One is overstimulation of visual, olfactory and auditory information, the lack of control in their environment to access resources that can meet their emotional and physiological needs, that reduced social interactions between species, like interspecies, and interspecies with, like, with humans most of the time. Then also the lack of predictability of daily events is usually a really big struggle as well. So with these challenges, this is how it affects their ability to regulate. So in terms of the TAPS, most of the time they're encountering quite protective bias emotions or that mixed emotion, and the arousal is generally quite high. So in terms of the protective bias emotions, dogs in kennels or confinement, frustration is a really big one. So if you know that a resource is available that could help you feel better and find relief, but you can't, it's not in your control just to go access it. So you have to ask someone or you don't know what time you get to go get it. That's usually a source of frustration. Any unmet emotional needs. So if you've got a dog that's got quite a high seeking or play need, and you can't meet it within this confined environment, generally frustration then comes to play. Then also they are kept for quite a long time, like we're talking overall 3 years of this environment. So if it's chronically, they don't have control to access and they're not feeling great because their emotional needs are not met, then frustration also comes into play over time. You've got fear and anxiety, which is, again, that unpredictable routine. Like, we don't know. So most of the time you would hope they know when dinner is and when they get to go out and etc. But there are invariably different routines because there's different staff, different things happen in the day. And sometimes it is quite hard for the dogs to know exactly what's going on. When you don't know what's going on, then you have questions of the what ifs, what does that mean? That contributes to anxiety. Lack of control of their environment. Again, if you don't know when you have control or access, that's also a what if what question. So you've got that anxiety driving component. Sensory overload. You don't know what this information means, but there's a lot of it and you can't process it. That's again to do with contributing to anxiety. Pain is one that is good to keep in mind, because with studies, it really depends on the research, but you do have like your veterinary procedures where you take blood or you sample tissues, etc. And you might have, if you have older dogs, then sometimes they do have some physical pathology that they might not say ow, or, you know, it doesn't affect overall what they look like and what they do, but they are experiencing discomfort. So then that adds on to the taps of that projective bias emotion. Confliction is when they have green and red emotions together. So, for example, they go, oh, I really do like humans. I like enjoying the company and whatnot. But then when sometimes they do things that I don't like. So then you have two together when you say the same person, and that's when you have a higher arousal. And mixed feelings is always worse than just one or the other. So that's something to be mindful of, is confliction. Second component of how it affects everything would be drainage component. We'll talk about how hard they'll find it to drain within this confined environment. So there's quality and duration of sleep. Again, it's to do with that kind of sensory overload and increased disturbance of sleep, or if you haven't clocked up the hours that you should be doing according to your life stage. Decompression activities, if there's insufficient things to do so in relation to the stress they encounter during the day, then sometimes drainage would be compromised. If they don't have a relationship to kind of as a backup support for their resilience, so then again that becomes very hard. So now we know what challenges they encounter in terms of emotional regulation and maintaining their emotional health. Then this is where I'll try and talk about how certain housing and enrichment can be used as tools to help these dogs emotionally manage and maintain their emotional health. So in terms of, again,
sorry, Findy.
Are we going over time?
We're a bit over time.
Yeah.
If you wouldn't mind wrapping up quickly, I'm really sorry, because
I will. That's fine.
Important, but
yes,
thank you.
No worries. I'll fly through this. So we're turning down the taps. So they're all pretty similar in terms of what they do if you think about it. So housing would be if you have physical barriers for that visual stimulation, you have music to calm down the auditory stimulus. Anything that's like non-slip floorings and coverings, bedding usually really helps as well for the feeling of security. Having a predictable routine is also very important. So we like to follow the eat, play, decompression, sleep routine. And then, as I said, predictable human interactions and relationships are quite important. So really good to just limit the exposure to the number of handlers, so you really get a good opportunity for these dogs to build a relationship. Once you have a relationship, there's two really good things. Is you get resilience. It's like a buffer for stress. And you also will find things a lot more predictable because you only, interacting with one person. Cooperative care was another thing that I was going to go through, but it's a very interesting topic because there's quite a few things that, in terms of benefit, to reducing emotional arousal and helping build that relationship. So I can put a link in if anyone's interested, just so they can read a little bit more about it. So what we're trying to do with cooperative care is really trying to teach them with procedural handling as a game, and these are the benefits. So they'll be more engaging when this procedure happens. You increase their choice because they choose whether they want to be in it. They can choose when it stops as well, and it increases the predictability of what they'll encounter in terms of if you have to sample tissues or take blood or whichever. So I'll put a reference in there for you in the link. The other thing is you can, with enrichment, what you're trying to do is you're going to increase that engaging bias emotion. So at least day to day, if they have more of these things, you'll have lower arousal through the day, then you're less likely going to dysregulate. And the second thing is that everything that we said before with the housing, with predictable routines, with cooperative care, with procedures, you're trying to reduce those protective bias emotions. So less goes in the sink. Then the last thing would be drainage, which is, again, if you have really good housing and you reduce that stimulation with various barriers and you have a feeling of security, you will increase that quality and duration of sleep. Especially in your routine. Usually we find that shelters that encounter, or like dogs in confinement, kenneling, if you have quiet time, even if it's just an hour in the middle of the day where there's no human activity around, generally dogs will have that time at least to drain whatever happened in the morning instead of building it up throughout the day till the end of day, and then the afternoon becomes awful. We like to schedule in what I call fruit breaks, like in little kids, but for dogs, you have these things just so they have little decompression opportunities on top of that quiet hour. So it really drains out everything. And then again, that building of relationship. There are challenges within it, because I understand that's just reality. But this is a very good paper that I found that's quite recent, and it illustrates all of that, of what you can do in terms of housing and enrichment for research dogs. I just wanted to use this opportunity to apply it to emotional regulation. So if you do use enrichment, it's then more purposeful and we know what we're trying to achieve. I think that's it. That's it. These are the references, but I'll go through a link with cooperative care for anyone that is interested. Sorry.
Thank you, Findy. Sorry to rush you because it was so interesting. Really excellent presentation. Such a clear introduction to the concept of emotional health. I know I personally find this sync model just incredibly helpful for understanding how animals feel. Please remember, if you have questions for Findy, put these in the Q&A and we'll come back to them at the end. And now I'll move right along to introduce our second speaker, Adjunct Professor Joanna Makowska. Joanna is Adjunct Professor in the Animal Welfare Program at the University of British Columbia in Canada. She's also Director and Senior Scientist, Animals in Laboratories Program at the Animal Welfare Institute, which is an animal advocacy organisation based in the USA. Joanna has a particular interest in laboratory rodents and learning how to give them a good life. Over to you, Joanna.
Just noticed I was muted. Hi, it's a pleasure to be here. I'm going to talk about the benefits of more naturalistic housing for rodents in research. So I'm going to start with outlining some of the issues that we do have with current cages. I don't really know who the audience is, but for those who don't know, the typical cage for a rodent pretty much around the globe is a cage that's the size of a shoebox. Problems with that is people started writing about this. And so in 2010, there was a nice paper in the Proceedings of the National Academy of Sciences that was quite controversial. And it said, control laboratory rodents are metabolically morbid, why it matters. And what they said in the paper is that failure to recognize that many standard control rats and mice used in biomedical research are sedentary, obese, glucose intolerant, and on a trajectory to premature death may confound data in interpretation and outcomes of human studies. They also said that if you're using these control animals housed in this way to test the effects of drugs, what you're really testing is the effect of a drug in a sick body and not one that is normal and meant to mimic or model the human condition. Another more recent paper that was published last year was titled Conventional Laboratory Housing Increases Morbidity and Mortality in Research. In this study, they found that conventional rodent housing increases all-cause mortality, so just dying for any reason, and also the severity of 5 diseases that are known to be exacerbated by chronic stress. So what this showed is that these shoebox cages aren't only making the bodies not normal, making the animals physically sick, but they also do suffer from chronic stress and are more likely to become sick and die of cancer, cardiovascular disease, and some others. Next logical question in my mind is, well, why are we using shoebox cages and where did they come from? The reason is it's just kind of pure historical coincidence. Back in the early 1900s is when we started keeping rodents in colonies and cages, and people were just here and there custom making their own cages. Everybody came up with something similar, and it was really just based on ease of handling and cleaning. And I'm going to leave you here with a few examples of pictures of what these cages look like. They're pretty much the same size and shape today. The only thing that's really changed is we've updated the materials. We used to use wood, then metal, and now plastic. And so basically the design that we're using today is based on tradition rather than scientific evidence. And so we need research into rodents' behavioral needs in order to inform appropriate housing conditions. So I'm going to describe first a study that I did for my PhD. I was just curious to see what rats would be like and how it would impact their welfare if they were housed in semi-naturalistic housing. On the left, you have a photo of a standard housed pair of rats, which is 2 rats with a little piece of tunnel. On the right-hand side is the cage that I used, which is a popular pet-style cage. It is very large. It allows rats to climb, and I also put burrowing substrate in the bottom in case they wanted to do that. I got newly weaned spag dolly rats from Charles River, Canada, which is one of the main suppliers of research rodents. And when the rats arrived, they were randomly assigned to each housing condition. Here, I'm going to show you a video just to compare how differently these rats behaved. I was panning, the rats are housed in the same room, so I was really just panning the camera from one side of the room to the other. And I just want to show you how different they are. People who work with conventionally housed rodents usually say that they're sedentary, that they're lazy, they just don't want to do anything, and that's what rats are. And that is so untrue. And it is also untrue for these rats that are commercially bred for generations and generations to be housed in these cages. They don't do anything because there isn't anything to do. There's no space. So in the study, my rats were continuously filmed with infrared cameras, and then I scored 3 behaviors that looked to be very frequent in these semi-naturalistic cages, but weren't even possible in the standard cages. Those 3 behaviors are burrowing, climbing, and upright standing. And for the purpose of time, I'll just show you burrowing and standing today. What I mean by upright standing is like full on back leg extended and the back is straight. Because in a standard rat cage, which is about 18 to 20 centimeters, the rat can't even put the back straight. And I scored these behaviors when the rats were 3, 8, and 13 months old in order to look at differences as the rats matured and grew up. I'm going to start with the first behavior, which is burrowing. And I'll start with a little video just for you guys to see what that looked like. In this case, the rat was burrowing right behind the plexiglass that was holding the soil in, so we get to see what that looked like. And I just want to say this is during the dark phase, so that's when the rats are active. The orange glow is just the type of lamp that we use that is beyond what rats can see, so for them it appears dark. So here's the first graph of the results. On the x-axis here, you have the age of the rats, 3, 8, and 13. On the y-axis, you have the daily frequency of the behavior per rat. What we found is that each rat burrowed about 25 to 30 times per day, and that was not significantly different as they grew up, which means that they continued to burrow at the same rate as they got older. This constant rate suggests that this behavior is particularly important to rats. And the reason I say this is that rats, such as and other mammals and humans as well, we all become less active as we get older. So picture like kids, they have trouble even sitting down for dinner. They'll eat quickly, then they're get up and run around the room. You have so much energy, you just do a lot more. And as you grow older, you become less active, and you're going to trade behaviors that are less important to you for rest, and things that are still very important to you are going to keep doing at the same rate. So what we found is that burrowing was one of these behaviors that really they kept doing at the same rate. Upright standing is the second one I want to focus on now. What we found is there were 2 types, and I'll walk you through them in the video. What you're seeing now is the first type. We call this an exploratory upright standing. What you see of the rat is fully extended. Her head is moving. She seems to be sniffing and investigating what's going on in the upper sides of the cage. And we're about to see the second type on the upper shelf. Here, this is a stretch. So what you see, there's a yawn, there's a paw stretch, and you're going to get to see it right in the middle again, yawn and paw stretch. What we found is that stretching was way less frequent. It was about 10% of all the uprights that we scored. Here are the results. You have the same type of graph. On this axis, you have the age of the rats, and on the other axis, you have the daily frequency per rat. What we found is upright standing was by far the most common behavior of the three that we scored. Rats were standing upright about 175 times per day, which is equal to every 4 minutes while they're awake when they were young, and still about 75 times per day or every 10 minutes when they were older. So this is something that decreased with age, was still quite frequent even when they were older. So, looking at this video has got me thinking that, you know, as I said, rats in standard cages cannot stand upright, which means they can't stretch this way. And so that led me to hypothesize whether, because they can't stretch upright, would they be doing a lot more of this lateral stretching than the rats that get to do both. And so to look at that, I compared the frequency of lateral stretching between the two systems. So the control rats and the semi-naturalistic when the rats were 13 months old. Here are the results. You have the housing condition on the bottom here, semi-naturalistic and standard. What we found is that indeed standard housed rats stretched about 6 times more when laterally. That seemed to be quite a big of a difference. And so then I looked at the amount of all stretching in both systems. Remember, in the standard system, they couldn't do upright. So this is all just the lateral. In the semi-naturalistic, the bottom bar is the lateral, the blue bar is the upright, and still the standard house rats are just, in general, stretching about 3 times as much. Again, that seemed to be a really big difference, and so it seems to say that the inability to stretch upright does not alone account for the high rates of stretching that we're seeing in the standard cages, and that something else is going on. If you look at the literature on stretching and why we do it, what the function is, it's a corrective response to stiffness caused by positional stress or immobility. And so the implication here is that low mobility in standard cages, so this includes just the lack of space, lack of movement, including upright standing and stretching. All this may lead to stiffness and positional stress, which then the rats attempt to alleviate through frequent stretching. So the conclusion from this study is that standard housing for rats is preventing them from performing a variety of innate behaviors that they are motivated to perform, and that shoebox cages are cramped. Now, for anybody who works in NAMS, you'll know that changing how you house these animals is hugely expensive. It is a huge endeavor. There's a real reluctance to change. It is in the hundreds of thousands of dollars. So what people have started doing is using playpens. The concept is that you have your animals still living in the shoebox cages, but they get regular intermittent access to a naturalistic area called the playpen. So in this study, we wanted to look at the effects or the welfare benefits of playpens for laboratory mice. We housed our mice in 3 strains per cage, so easy for easy identification. And we gave these mice regular access to a playpen. We built our playpen in rat cages that are much larger for little mouse. And one side of the playpen had burrowing substrate, and the other side just had fun toys, things to climb and run. Our mice were given voluntary access to the playpen, so we bring their home cage close to the playpen, and then they had the choice to use the tunnel and cross over. Here's a video just to show you what they did. And I'll point out that unlike the rat videos here, the animals, this is during their daylight, so while they should be sleeping, and yet they were still quite playful and active. All right, so one way that we looked at whether mice enjoyed this experience or whether it improved their welfare is to use something called anticipatory behavior. That's a behavior that is seen. You'll recognize it, for example, with your dog. When a cue signals that something fun is about to happen, the period between the something fun and the cue is this anticipatory behavior phase. So for example, if always when you grab your keys or you grab your dog's leash, that signals to your dog that you're about to go out, you'll see they'll get up, they'll stop moving, they're wagging their tail, they're very active. So in this study, we used a cue of fingers across the lid that made a sound. You're about to see that. In the control treatment, we made that cue, the mice waited for a minute, and then their cage just went back on the cage rack. In the playpen treatment, we did the cue, waited a minute, and then they got to go in the playpen. So as you can see already without actually seeing the quantitative results, is that the control mice are kind of not doing much. The signal doesn't really mean anything to them, whereas the other girls are very active. So this is what we call their anticipatory behavior period. We quantify anticipatory behavior by looking at how many different behaviors they do. So you'll score every time they change behaviors. All right, so here is the graph showing you the results. On the x-axis, you have the three different strains that we used. And then on the y-axis, you have the change in anticipatory behavior. So how many behaviors per minute they did from baseline. And what you see is in all three strains, the mice that went into the playpen were a lot more active than the control mice, showing us that yes, the playpen is considered or seen as a reward. What we also looked at is aggression. Mice can be quite aggressive, females less so than males, but it is nonetheless quite a big welfare concern. What we found is we looked at aggressive events following a stressful event, which is cage changing. Aggression always increases in the hour after they're put in a new clean cage. So what we found is that playpen mice were actually much less aggressive than control mice. So another positive experience, something about the experience of going into the playpen. Once they're back in their home cages, they're still a little more resilient to stress. However, we also found that these animals, the playpen mice, were actually performing more stereotypies in their home cages compared to control mice. That was not expected. Stereotypies are abnormal behaviors that happen in suboptimal environments. So we were hoping that this experience of going to the playpen, we did half an hour 3 times a week, would actually make them develop less stereotypies because they'd be just coping better. But we found the reverse. So there's a few potential explanations. It could be that once you experience something better, once you experience a playpen, being back in your home cage 23 hours a day or 23 and a half makes it even worse by having that contrast. There's some data showing that stereotypies, especially bar biting, might indicate escape attempt. So it's possible that these animals are just trying to escape even more so because they want to get to the playpen. It could potentially be a sign of active coping. What I mean is these playpen mice just were put in an environment where there's actually stuff to do. They can burrow, they can have an impact on their environment. And so they learned that they can do active behavior to try to fix things, whereas the control animals don't have that experience and they might be more likely to do this inactive but awake, so more depressive-like inactivity. So the conclusion from this study is that mice do want to go into playpens. It's something that they find rewarding. Playpens can reduce aggression after stressful event in the home cages, but it isn't still not sufficient to mitigate the development of stereotypies, and it makes them worse. So we're missing something. It's possible that playpens just are not enough, and we need more research. I'm just going to finish quickly, 30 more seconds, just give you one other more inspiring example of something even more naturalistic. This is an example from Dr. Potts at the University of Utah. He's housed his mice in what he calls barns. He has about 30 mice living in this barn, and there are territories. There's half of them are high quality territories, the other half are low quality. Mice that are released into this barn will compete for territories, and then the more successful ones will have more mates and produce more offspring. He has used this setup to look at drug safety testing. And he's found that it's a lot more sensitive than what we traditionally do with shoebox housed rodents. For example, this is just one of his papers. He looked at one drug, simvastatin, or a placebo. So half the mice were given the drug, the other half were given a placebo. What he found is the mice that were given this drug had 41% fewer offspring, occupied 63 percent fewer territories, and had 10% lower body mass. Interesting thing is that this drug is one that was deemed safe. It was tested in control rodents. It was tested in another large species. Everybody, all these research showed that it was safe. It was released to the human population and then had to be recalled because it was found not to be safe. And so he argues, had we actually used rodents that are housed more naturalistically and have better welfare, we would have been able to pick up these differences. Thank you.
Thank you, Joanna. What a fascinating presentation. I especially loved your videos. They're so powerful to see the differences. We've had a few questions come in about whether you will have access to PowerPoint slides and references from the presenters. We will send them out with the Q&A document. So yes, you will have access to those. It looks as though we're not going to have time to answer questions live today, but please still send your questions in through the Q&A function and we'll put those all into a document for you that will go out with the webinar recording. So now we're going to move on to the final pre-recorded presentation for the day, which is from Dr. Chloe Stevens from RSPCA UK. Chloe is a Senior Scientific Officer for Refinement in the Animals in Science Department at the RSPCA, the world's largest animal welfare charity in the UK. She works to promote refinements and improvements in welfare for animals used in research and testing, with a particular focus on fishes and other aquatic species. She has studied behaviour and welfare in a range of fish species, including zebrafish, guppies and mollies, and has a PhD from the University of Exeter in stress biology, welfare and mortality in ornamental fishes. Just to note that the sound quality is a bit patchy on this recording, so check your volumes right up, and please enjoy this recording from Chloe.
Good morning, everyone. Thank you for joining me for this talk, and thank you also to the organisers for the invitation to speak to you today. My name's Chloe Stevens, and I'm one of the Senior Scientific Officers in the Animals in Science Department at the RSPCA in the UK. And if you're not familiar with the RSPCA, we are the largest animal welfare charity in the UK and one of the oldest in the world. And in the Animals in Science Department, we work to reduce the impact of science on animals by working constructively with governments, regulators, industry, academia, and other NGOs in the UK, EU, and around the world. And we do this through two major work streams, the first of which involves working to ensure that systems of ethical review are robust and operate effectively. And the second work stream is promoting the fullest possible implementation of the 3 Rs. And my talk today forms part of our work on refinement. So, what is enrichment and why do we care about it for zebrafish? Well, by now, I'm sure that everyone is actually pretty familiar with enrichment, but just to give you the definition that I'm using for the purposes of this talk, enrichment can be defined very broadly as modifications that are made to the environment of captive animals with the aim of improving their welfare. And you can see some examples on the slide of enrichment for other laboratory species. And for zebrafish, this might include live food, social housing, gravel, pictures of gravel under tanks, real or artificial plants, and other tank ornaments or objects. But despite these options, many zebrafish labs may look similar to these, with tanks that are small and fairly featureless and don't appear to be particularly enriched. And you don't just have to take my word for this. An international survey that was conducted by the NC3Rs in the UK in 2017 looked at various aspects of zebrafish use and care, and they found that only about 25% of respondents were using enrichment in the tanks, although most did report that they were using live food. And this is despite the fact that a wide variety of benefits of enrichment have been seen across all kinds of different species, including impacts on growth rate, immune system function, neural development, reductions in anxiety, lower aggression, and less neophobia. Now, sometimes I hear people express concerns over whether these kind of benefits have been observed in zebrafish. But in fact, almost all of these benefits have specifically been demonstrated in zebrafish, and I'm going to discuss a few examples of studies that have looked at that in a moment. But the fact is that given these benefits, the fact that many zebrafish facilities seem to be using very little enrichment is a definite welfare concern. So why should we use enrichment then? Well, as I've just shown you, enrichment can have a wide variety of effects on physiology and behaviour that are likely to represent better welfare, and we have an ethical or a moral responsibility to try and provide the animals in our care with the best possible welfare we can. We also, many of us, will have a legal or a regulatory responsibility to improve welfare. This will depend a little on where you are in the world, but most of us will have some sorts of laws or regulations that we must abide by that have a responsibility to improve welfare embedded into them. And finally, a good reason is this old adage, which I'm sure everyone is very familiar with by now, which is that happy animals make good science. Animals need to have normal physiology and behaviour for us to get good, valid data from them. And to achieve this, we need to ensure that they experience good welfare. So, what impacts does enrichment actually have on zebrafish? So enrichment is often discussed in the context of these 5 categories proposed by Bloomsmith, and I'm going to use these today. And the category I'm going to start with is physical enrichment. So this is the category that's best studied for zebrafish, and so it's the one I'm going to spend most time talking about. And it refers to physical objects or items in the zebrafish tank, such as plants, rocks, pipes, or substrates. So a key question for any form of enrichment is, do the animals actually want access to that enrichment in the first place? And that's what this first study looked at. So this is probably one of the best known papers on this topic. And the authors housed zebrafish in these tanks with 2 chambers where the fish could swim between each. And each chamber contained a different condition. And they looked at percentage occupancy. How much time were the fish spending on the barren side of the tank compared to the enriched side? And what they found was that for several forms of enrichment, including gravel, sand, and a floating plant, the fish were spending much more time occupying the enriched side of the tank than they were in the barren side of the tank. And although this is perhaps one of the best known studies, as I say, about zebrafish preferences, it isn't the only one. There are several others which have found that zebrafish prefer structured environments, or those that contain physical enrichment. Now, in the next study I want to show you, the authors focused on the effects of enrichment on survivorship at 30 days post-fertilization. In other words, how many zebrafish larvae were surviving when they were about a month old, when housed with either gravel and a plant or in a barren tank. And what they found was that larvae which had been raised in the enriched tanks had much higher survival rates of over 80% than those raised in the barren tanks at about 55%. Also, after a couple more months, the zebrafish were tested in the novel tank test, which is a validated and commonly used test for measuring anxiety-like behaviour in zebrafish. And they found that anxiety was lower in fish raised in the enriched tanks as well. And again, here are some other examples of studies that have found physiological benefits from being housed with enrichment, such as lower stress, marked by lower cortisol after being exposed to a stressful event, which may indicate a better ability to cope with stress, less anxiety, improved learning, and reduced aggression as well. The last piece of work on physical enrichment that I want to highlight for you is from this recently published PhD thesis by Michelle Lavery. One of the first things she did was to study zebrafish preferences for enrichment such that they could be arranged along a scale of preference. And she showed that zebrafish preferred plants over an empty tank. They preferred gravel over plants. They preferred plants and gravel over gravel only. And they preferred more plants and gravel over fewer plants and gravel. So she then housed zebrafish either in their most preferred enriched conditions or in barren housing. And what she looked at was probability of survival. And she found that probability of survival was much higher for zebrafish housed in enriched tanks. In other words, zebrafish housed with enrichment were living longer, which I'd say is pretty compelling evidence that physical enrichment is beneficial for zebrafish. So I'm now going to talk about each of the other categories of enrichment, but as there are fewer studies in these areas, I'm going to be touching them on them a bit more briefly. So next I'll talk about social enrichment, so providing animals with other individuals to create a more socially complex environment. Now, presumably, I don't have to convince anyone that zebrafish are a social species. For example, we know that they live in groups in the wild. We know that when we give them a choice in the lab, they prefer to be near other fish. And we also know that they generally prefer larger groups over smaller groups. But is that the whole story? Well, there are certainly studies that have found that being in groups instead of being isolated is associated with better welfare indicators like lower cortisol, i.e. lower stress, and lower anxiety. But There are also some studies that have found the opposite. And in fact, there are some that I haven't listed on the slide that have found no effect of social or isolated housing on cortisol or anxiety. So what's going on? Why these conflicting results? Well, maybe it's that there are other factors at play here. Perhaps what matters is not the presence of a group, but that group's composition. For example, some studies have found that single-sex housing can improve several parameters associated with welfare. And although I'm not recommending single-sex housing specifically, as that comes with a host of other implications, it does support the idea that group composition matters to zebrafish. We also know that zebrafish recognize familiar individuals and interact differently with them. So maybe being with a group of familiar individuals is more relevant for welfare than being in a group of unfamiliar individuals. There also could be other factors at play, like the stocking density that was used in these studies, the welfare measures that were used, or a combination or something else entirely. So what I would conclude from all of this is that while zebrafish should be housed in groups, we do need to be aware that social dynamics are complex, and therefore so is social housing as a form of enrichment. So we cannot simply say that zebrafish housed in groups are in an enriched environment. I would say that providing company of any sort to your zebrafish should be considered fulfilling a basic need rather than providing them with enrichment. So if social housing is the only form of enrichment you're providing your zebrafish, I would strongly encourage you to think about how else you might be able to introduce enrichment. And as a broader point, we need more research into factors that will help us understand what a socially enriched environment looks like for a zebrafish. So let's move on to sensory enrichment. So these are enrichments that might involve visual, auditory or olfactory stimuli, and they often involve modifications that don't necessarily require putting something into the tank. Do we have evidence that zebrafish respond to these kinds of stimuli? Well, yes, we do. And to demonstrate this, I'm going to go back to a study that I've actually already shown you, which was this one with the choice tanks. So if you look at this image on the bottom right, you can see an example. In this condition, the zebrafish were tested for their preferences for an image of gravel over a barren tank. And just as with the physical enrichment, you can see that the zebrafish much preferred being on the enriched side over the barren side. So this shows that just the presence of a picture was enough for zebrafish to express a preference about the kind of conditions they wanted to be in. And in fact, if I put the graph for real gravel back up here, you can see that the strength of preference for a picture of gravel was almost as strong as for the real gravel. Another study that looked at the effects of visual enrichment found that images of a seascape on the sides of the tank led to higher egg production over tanks with bare walls. And visual enrichments could also include things like the tank colour. For example, this study found that zebrafish had lower anxiety when housed in blue or black tanks over white or transparent tanks. And a form of enrichment that's been proposed, but unfortunately not directly tested as far as I'm aware, is the inclusion of dawn and dusk phases in the lighting cycles that are used in the lab. And the idea of this is that it can reduce stress that might be caused by the startle response that might be seen in zebrafish exposed to sudden changes in light levels. Now, sensory enrichment doesn't just have to be visual. It can be in other modalities as well, including via sound. In this study, the authors housed zebrafish either in tanks exposed to the ambient noise of the lab or exposed to classical music. And they tested their fish for anxiety in the novel tank test. They placed them in a novel tank and they looked at their behaviour. And they found that in many of the measures, the fish that were exposed to the music showed behaviours which are associated with lower anxiety levels, such as more time spent, greater distance travelled, and more time spent mobile in the top of a novel tank. And once again, this is not the only study to find effects of auditory enrichment. This result was replicated in another study a couple of years later. The next type of enrichment I want to highlight is dietary enrichment. And in this case, because laboratory animals should ideally be provided with a complete diet, I'm not referring to anything that has any sort of nutritional benefit, but rather enrichments that may improve welfare through some sort of benefit external to the animal, such as through providing variety. And for zebrafish, this would most likely be through the provision of live food. Live food is usually suggested to provide opportunities for displaying natural behaviour and perhaps to alleviate boredom. But despite having looked many times for studies that demonstrate a welfare benefit, I haven't been able to find any empirical evidence for this. And if you know of a study like that, then please let me know. Now, this isn't to say that live food isn't enriching. I think it probably is, and I would encourage people to use it. But It's interesting to me that it's commonly described as an enrichment, and yet enrichments for which there is much more empirical evidence are much less widely adopted. So I do think it gives us reason to reflect on both our own practices and the level of evidence that we are willing to accept or that we feel that we need to introduce some kind of beneficial modification. So the final category of enrichment is occupational enrichment, and this refers to enrichments that give the animals something to do. In other species, it might refer to things like toys or puzzle feeders, and it's a little bit trickier to think about how we might do that for zebrafish, but there might be ways that we can do this. So one way that might work is by giving zebrafish water flow to swim against. So in this paper, zebrafish were placed in a multi-chambered choice tank, and they were given the choice of a barren or plain compartment, one that contained water flow that they've called swimming only, one that contained plants and substrate called enriched, and one that contained both water flow and plants and substrate. And on this graph, bars above the line show a preference for that condition, and bars below the line show an aversion. So you can see that both the barren chamber and the water flow on its own were aversive to zebrafish, but water flow combined with physical structures was the most preferred condition, even more so than just physical enrichment on its own, suggesting that water flow might be valuable to zebrafish and something that they do want access to, but with caveats, perhaps only when physical structures are in place to break up the water flow. So really the key message for me is that there is good evidence that it's possible to provide enrichment that improves the environments and therefore the welfare of laboratory zebrafish. And although we probably haven't established the ideal conditions, there are some modifications, there are some options that are relatively easy to implement and likely to benefit the fish, such as using gravel images under tanks. Or even if you can't implement a modification in all tanks, you may be able to do something like provide artificial plants if fish have to temporarily be housed singly or in pairs for various reasons. So I do encourage you, if you work with zebrafish, to consider trialling new enrichments and interventions in your facilities, but I encourage you to do so in a scientifically robust way, and then to please publish or disseminate those results so that we can all learn and benefit from more collective knowledge. Now, the studies I've covered in this talk are just a subset of those that we reviewed for this paper, which is published open access by the journal Animals, which you can get to through the tiny URL link on the slide. And for that paper, we combed the literature for every paper we could find relating to the effects of enrichment on zebrafish. And the results of those papers, including the type of enrichment studied, the welfare parameters used, and the effect of the enrichment, are all summarized in easy-to-read tables like these. So do feel free to take a look if you're interested. And I just want to finish by highlighting some of the other resources on zebrafish refinement that are available on our website. You can also see our Twitter handle on the slide. And if you'd like to hear more about any new resources or events that we run, there's also the link to sign up to our quarterly newsletter as well. Thank you very much.
Thank you to Chloe for another really interesting presentation. It's a shame that Chloe couldn't be with us live today, but we really appreciate her doing that recording for us from the UK. That brings us to the end of the webinar. Thank you for your attendance today and for sticking with us right to the end, even though we've gone a little bit over time. Thank you especially to our wonderful speakers for giving us all lots to think about. What fantastic presentations. Findy and Joanna, if you're still around, can you turn your cameras back on so we can give you a big virtual round of applause? Thank you. It was wonderful. Hopefully everyone attending has found something in today's presentations that you can take away and apply, even if it's for your own non-human family members at home. And thank you especially to those who have submitted questions into the Q&A. You probably still have a few minutes to jump some more questions in there if you would like to. We will follow up in coming weeks with a recording of the webinar and answers to all of your questions. Keep an eye out also for the next Animal Research Review Panel webinar, which is scheduled for November, and you'll receive more information about that one by email closer to the date. So thank you again, and we look forward to seeing you all at the next webinar.